Right. You spoke there, didn't you? Or was that 2 years ago that you had a little
Yeah, yeah,
breakout session?
yeah. I did a breakout session in Tri County. That's right.
Right.
Much how many scientists and how much work is being done in Ohio. Really enjoyed Ohio State University presentations in spring and in fall. There was a virtual meeting, I think.
Yep. Yep.
Yeah, lots of really good beekeepers. We're not beekeepers by training. We became beekeepers about 5, 6 years ago. Um, we were biologists and interested in um, probiotics for animals that are called direct fed microbials. And we were studying that and working on that
Yep.
before we became beekeepers.
Okay.
And that was a fun journey. I really appreciate coming from little knowledge of beekeeping and um, learning everything and keeping the bees the first year, the second year. What kind of I guess membership do you have? Is there a lot of new beekeepers, beginning beekeepers, experienced beekeepers?
Well, if we were meeting in person, it's usually about beekeepers about the first 3 years.
Mm-hmm.
So, and it's hard to tell online of who's who anymore because most of the people shut their cameras off. I know, and they just have a first name. So most of the people are going to be somewhat experienced beekeepers, or at least got one season under their belt.
That's good to know. And I would be happy to focus on things and questions that you have, specific things that you are interested in.
Okay.
Obviously, I mean, for us, we'll talk a lot about probiotics. I would like to talk about Super DFM and how that
Yep.
came
Oh, yeah.
around to me. Um, but I thought I would start out, now it's probably a good time to start. I thought I would start out by just talking about microbes, bacteria, what they are, why do they even matter, especially to beekeepers.
Okay. Yeah.
And so for many, many hundreds of years, we didn't know about microbes and bacteria and what happens when our dough rises and bread ferments, right? Lots of magic happening there.
Okay. Well, I'll introduce you since it is 7 o'clock. We might as well get the show on the road. Welcome everyone to the Maumee Valley Beekeepers February meeting. I told my wife today, I've been president for 15 years, and if we would have been holding a meeting in person, today would have been the first time we would have ever canceled a meeting because of snow. So that's kind of miraculous that I've gone this long without having a big snow event on a meeting day. I thought it was just the magic of Maumee Valley Beekeepers meetings that it never snowed, but today was the day, but we're virtual anyway. So we're gonna, I'm glad I invited several clubs. I see we have some non-Maumee Valley beekeeping members. Welcome, welcome. We're glad to have you. We've been invited to other clubs, so I figure I'll extend the olive branch to everybody else, and we'll enjoy each other's meetings. Tonight we have Dr. Vera Strogova, a scientist fascinated with microbes. She's a co-founder and CTO of Strong Microbials. Vera is charged with the development, research, and improvement of probiotics for honeybees. Vera's work builds on her experience as a quality and safety leader and research scientist working on animal probiotics in Daninsko DuPont subsidiaries. Vera earned her MS in biology from the University of Wisconsin-Milwaukee, where she studied how organisms respond to sugar, and a PhD through Marquette University, where she studied new aspects of how cells breathe. Vera is currently serving on the American Bee Federation Research Committee, the Colossus Nutrition Task Force. So with that, I'm going to introduce Vera. We are having a few audio glitches with Vera's microphone, but it seems to be doing pretty good right now. So, and for all of us people in Ohio that got snowed out of work today, Vera had to go in and grow some microbes, she told me this morning or earlier in the meeting. So Dr. Vera, it's all yours.
Thank you, Roger. Thank you to everyone who is thinking about their bees today. And I really admire and appreciate and respect beekeepers for their commitment to learning, even when it's, not the coldest of the winter, not really a season to keep bees. I find that beekeepers really use all of the off time that they have without bees to learn how to keep bees better, how to make them healthier, and just understand all the processes in the hive, from queen rearing to feeding and nutrition. So it's really fun to talk and be part of many beekeeper meetings. I wanted to start off with just general introduction of what do microbes and probiotics have to do with beekeeping, and why should we care? So for hundreds of years, for thousands, for millennia, we didn't really care or understand the microbes that make different processes, like from digesting our food to fermenting the dough in the bread. So we just kind of didn't know what was happening and didn't appreciate and feared the microbes that cause diseases and infections in people and in animals. We're now really at a time where we can understand and appreciate the complexity of microbes in the backyard, in the dirt, in the pond, in the beer or bread that we cook and prepare using microbes, as well as in the beehive and the things that are happening inside the bee gut. So that's what I dedicated my life to, is understanding these really great helpers, microbial living organisms, which are a great tool for beekeepers and farmers and actually all of the humanity. And we, with my husband Vera Strogova, we founded Strong Microbials in 2012 to grow microbes and to use and produce them to improve agricultural processes so that the agriculture is cleaner, more efficient, and we can use the microbes to both save money to beekeepers and farmers, and also to make sure that we don't have synthetic chemicals, or minimize the synthetic chemicals, antibiotic and pesticide use in agriculture, because that really pollutes not just agricultural products, but also our environment. And I think that beekeepers are probably of people who are most aware of their environment and how little changes could lead to big impacts on bees and beekeeping. Right, so I wanted to talk about honeybee microbes. Which microbes do we know? Which ones are good or bad microbes? I want to talk about the bad ones in particular because there are obviously bacteria and microbes that we can all think about that cause microbial infections of honeybees. For many years, the go-to way of treating infections of honeybees was to use antibiotics. We now are more and more aware of regulation for antibiotics and the bad consequences of using antibiotics. So to start off, I wanted to talk about what is already known about bees and microbes and antibiotics. This is a picture that brings me much joy. So when we see the nurse bees feed honeybee larvae, that is an excellent way for bees to protect the next generation from pesticides, to provide balanced nutrition. So nurse bees feeding the honeybee larvae, probably, agree with me or not, if you can hear me. Let me know what you think. I think it's one of the most important things that happens in a beehive. And it's important because it actually lays the groundwork for the next generation of bees, because those bees need to be fed by the nurse bees. And when you can clearly see when that's maybe not a first thing that you'll learn as a beginning beekeeper, but once you have a few years under your belt, you can tell right away if there are problems with the hive and when there's not enough nurse bees, because you can see whether the larva looks good or not. So you can tell that on the left side of the screen, there are larvae that are covered in worker jelly, that are floating, that are wet, that look good. And you can tell right away if there is not enough nurse bees or a nutritional problem. Sometimes it could be an infection too that will decimate the number of the worker bees and the nurse bees in the hive. You could tell right away when there's a dry larva, and that's not something that you want to see when you're expecting the hive, right? So those nurse bees feeding the next generation of bees is so important. And it's something that maybe we take for granted unless it doesn't work. But who is feeding the nurse bees? Of course, they feed themselves. The nurse bees would partake of bee bread and honey and feed themselves, really. But there are also lots of different bacteria in the bee bread and different microbes in the bee bread and in the honeybee gut, as well as in the pollen and nectar that bees consume and harvest. And all of the bacteria and microbes in the hive, on the comb, and in the gut of the honeybee. And the gut of the honeybee could be divided into kind of the part of the gut that's closer to the head, so that's proximal, and that's crop and ileum, and the part of the gut which is closer to the rear, and that's the rectum and the midgut and the ileum and the rectum. And so those, as you move through the gut of the honeybee, there is a lot of different bacteria that are lining and populating the gut of the honeybee. And these microbes and bacteria actually help to feed the nurse bees because they digest the food that the nurse bee eats. Especially in the rectum and in the hindgut of the honeybee, there is a lot of bacteria that belong to the lactic acid bacteria group. So they are in the family Lactobacillus and Bifidobacterium. And in a single honeybees, there could be billions of lactic acid bacteria in their gut, which serve many very important functions for the bees. One of the most important functions is to break down or promote digestion, breaking down the food molecules that the bees consume. These bacteria are very important for digestion, and that's why I call them, or I say that these bacteria feed the nurse bees, because having adequate nutrition is important for all bees, but especially nurse bees. And I just had a little sign that said my internet connection was weird. So I just want to check that you can still hear me all right.
Yes, we hear you just pretty good. It kind of fades in and out a little bit, but you're good.
Thank you. Yeah, so I just wanted to bring you into the gut of the bee, and there's bacteria in there, right? And those are important. So there's a little bit more kind of science behind the gut bacteria. So these gut microbes, they help digestion. So bacteria like Lactobacillus that I was talking about and other ones break down glucose and fructose into smaller molecules like succinate, propionate, butyrate, acetate. Those are so-called short-chain fatty acids, and they're shown in this blue square in the middle. So that blue square represents the gut, and those octopus-looking things are the gut lining. So the short-chain fatty acids that the bacteria produce, when they break down other molecules in the bee nutrition. Those easily cross the gut barrier into the hemolymph, providing readily available energy for the bees to do their work. And studies, including this one, which was done in 2017, but that's not the only one. There are many studies now that show that not having proper number and diversity of microbes in the gut, of bacteria in the gut, or not having the lactobacillus, which are the lactic acid bacteria in the gut, shows that they play a role in weight gain, so the bees growing and getting bigger, as well as sucrose response, which is their response to food, their appetite, and other parts of physiology which ensure normal development, such as vitellogenin, which is the protein which makes up majority of the fat body of the honeybee. All of those are very important physiological functions, and they all depend on proper nutrition, which starts with gut microbes. So actually, one of the methods that the scientists used to study the importance of gut bacteria was to use antibiotics to wipe out or remove gut bacteria. And when they did that, they could tell the difference in weight gain, sucrose response, fat body, and other physiological functions of the bees. Okay. So nurse bees need to eat, and they need the gut bacteria to get proper nutrition. Nurse bees feeding the larvae is very, very important because larvae are susceptible to so many diseases, and many of these are microbial infections of honeybees. We know, for example, American foul brood, which is caused by a bacteria. It's a bacterial infection, AFB, that's caused by Phenobacillus larvae. And P larvae could be treated in their vegetative state by antibiotics. So they could be killed by antibiotics. Although for AFB, veterinarians or apiary inspectors do not actually recommend using antibiotics to treat an active case. That's not the only and not the first solution to treat an active case of PV. So bacteria P larvae is susceptible to antibiotics. So is the bacteria that is causing European foul brood or EFB. That's a bacterial infection caused by a different bacteria. That bacteria called Melissococcus platanus could be killed and is susceptible to other antibiotics. So some of the microbial infections of honeybees could be treated with antibiotics. Sac brood and chalk brood could not be treated by antibiotics because they're not bacterial infection. Sac brood is a disease of the brood that is caused by virus, and viral infections do not respond to and cannot be treated by antibiotics because antibiotics only work against bacteria because they have a specific mechanism of action. The chalk brood also is an infection that affects brood, and you know that all of these brood diseases, foul brood, AFB, EFB, sac brood, chalk brood, and that doesn't even cover all of the possible diseases of the brood. They all affect the developing larva or the brood of the honeybee. Part of the reason for that is that the brood of the honeybee or honeybee larva are so vulnerable because they actually lack the abundance of normal bacteria in their gut during their development. And they depend on the nurse bees to be fed and taken care of until they become an adult and develop their own microbial gut microbes, gut bacteria, microbiome of the gut. So chalk brood and sac brood are not infections that could be treated by antibiotics. And currently, because of that, there is no treatment for chalk brood or sac brood. I know that I'm not covering in this slide all of the different infections of the brood. Is there something specifically is there another brood disease that you're thinking about? Maybe a virus? something that you see on a regular basis?
I don't see too much. We've had some instances of European foul brood in our area.
So these are different microbial infections of honeybees, and there's another one that affects adult bees this time, not the developing bees, not the brood, but the adult bees. And that's nosema, and that's something that we see a lot of times too across in different regions, and it's a seasonal disease, so you see a lot more nosema in the spring than you do in the summer. Nosema is a fungal infection. It couldn't be treated by the antibiotics that are used for AFB or EFB prevention, but they could be treated with antibiotic that is called fumagillin or fumagillin B. And so using the antibiotics to treat some of these microbial infections is not something that you or I really want to do, but in some cases, there is just a very strong disease that needs to be treated with antibiotics. There are some downsides to using antibiotics, although there is also an advantage, because using an antibiotic will eliminate the susceptible bacteria. Using antibiotics, of course, depends on diagnosing the disease correctly. And in case of EFB, for example, or nosema, there are diagnostic tests that beekeepers can use very easily. Or in other cases, those bees could be sampled and sent out for analysis to know what the nosema loads are. But there are additional problems with antibiotics. Using antibiotics right now for beekeepers requires a veterinarian prescription, which means that a veterinarian has to come out and diagnose the infection correctly. Historically, many beekeepers didn't work with veterinarians, and many veterinarians are not very educated about bee diseases. The reason why using antibiotics without veterinarian prescription is regulated and it's not permitted is because prophylactic use of antibiotics, prophylactic is using the antibiotic to prevent a disease that you don't have. A prophylactic use of antibiotic, for example, is using the antibiotic, taking an antibiotic before doing some dental work, which is something that many people are familiar with. In this case, you're taking antibiotics before there is a risk of infection to prevent an infection. And that's something that in human medicine could be done, but in veterinary medicine, including beekeeping, prophylactic use of antibiotics needs to be regulated, at least that's the understanding right now, because it's very widespread. And widespread prophylactic use of antibiotics leads to resistance to the bacteria that can no longer be treated and killed with antibiotics. And that's what leads to multiple drug-resistant infections, which cause serious problems for humans. And that's what started the regulation of use of antibiotics in all of the agricultural animals, and only since 2016 or 2017 in the United States was also started including beekeeping antibiotic use. We also know that using antibiotics can contaminate bee products, honey and beeswax. And we also know that using antibiotics will disrupt gut microbes or gut bacterial community and increase the susceptibility to other infections such as nosema, because using an antibiotic for EFB is not going to eliminate nosema, which is not susceptible to that antibiotic. So that's just kind of an introduction to bacteria and antibiotics and their use in beekeeping. I wanted to spend the second half of my presentation talking about honeybee probiotics. We know that for many years, there's a lot of studies that show different benefits of using probiotics, feeding probiotics to honeybees. We have been also doing some field trials to look at digestion questions and the weight of the honeybee, as well as protecting the honeybees from infections. Normally, whatever bad or pathogenic microbes exist, so they're the purple ones and the green ones in the right part of the screen, those could be outbalanced by beneficial microorganisms. Some of the beneficial microorganisms are native or exist in the honeybee gut to begin with, and others could be probiotic. So they come from bee bread, they come from feeding a probiotic, they come from pollen and nectar, as well as the gut and the hive environment and hive materials. So ensuring that there is enough good beneficial microorganisms, such as the lactic acid bacteria, which I mentioned, and I will talk about them again, ensuring that there's a lot of beneficial bacteria all the time is one of the ways to make sure that there's not an infection, making sure that they outbalance the pathogenic bacteria. At Strong Microbials, we wanted to listen to potential problems in beekeeping and agriculture and understand what are the different problems and how can we solve them. So in 2014, we were approached by Eastern Agricultural Society master beekeepers, Earl and Carol Hoffman, and they had some issues with their bees, some problems that we were able to solve. We were interested in this probiotic approach to honeybees, which hasn't been practically done before. There was a lot of research, and part of that research was a study that looked at colony collapse disorder in early 2000s. In 2007, the study did a metagenomic survey of all microbes in the honeybee hive, and they were looking for a guilty microbe. They were looking for a microbe that would cause the CCD. And those of you who have been keeping bees at that time would remember that there was a lot of viruses and different microbes that were thought to be maybe guilty of the CCD, but they didn't find a particular microbe in this study that could cause the CCD. One of the interesting things that the analysis of healthy non-CCD colonies and not healthy CCD colonies was the insufficient bacteria called lactic acid bacteria. In the pie chart, it's shown by the orange and red colors. And you can see that the healthy hives have had much more lactic acid bacteria compared to the unhealthy hives that collapsed. That was one of the first kind of directions when we wanted to help the honeybees that Earl and Carol had. We looked at different studies that were done in early 2000 and found that there was a number of beneficial of lactic acid bacteria and bacillus bacteria, which are safe to use, have been fed to bees, and these are the different studies where they actually fed these bacteria to bees and found that they have one or another benefit in the laboratory study or in the hive study. We selected 7 bacteria based on both their safety record, general knowledge, general use, because some of the lactic acid bacteria are also used in other kinds of probiotics. So they have been manufactured and used safely for decades. And we also knew that starting with, I think, Evans and Lopez, 2 USDA scientists in 2004, that feeding these probiotic bacteria to bees is going to be safe for the bees, and we will elicit the good immune responses and promote digestion. And they were also known to inhibit or prevent some of the pathogens from occurring in the honeybee gut. I don't know if this video will play, but this is a video of Super DFM that we developed for honeybees and the bees consuming or eating it. Obviously, an important consideration when developing a new nutritional supplement for the bees was that the bees would like to eat it. And it's very important to feed this probiotic in a dry form because lactic acid bacteria are anaerobic, and thus just diluting them in sugar syrup or water will make them not last long enough for the bees to finish eating the pail of syrup or the feeder pail. And that's why we recommend feeding it dry, and the bees really like to eat it that way. And of course, we didn't want only for the bees to enjoy eating the probiotic. We didn't want to only rely on the scientific data that was oftentimes a laboratory study in a cage. We wanted to go to real beekeepers like Earl and Carol and dozens of others beekeepers that we worked with during the years to see whether there are practical benefits to using probiotics in beekeeping. One of the first things that we noticed was positive results that Earl and Carol reported to us. They had a couple of really rough winters before we started working together, and they split their beehives into two groups. They had a negative control group, shown here in blue, which didn't receive the probiotic, and the other group of hives, they had Super DFM probiotic before overwintering. They reported a 10% better overwinter colony survival, which was very important at the time and still remains very important for many beekeepers. One of the most all-inclusive measures of beekeeping success is overwinter colony survival. I also want to show that bee-informed partnership measures overwintering colony survival in every state. And that year, overwinter survival that was reported in the state was even lower. It was above 60%. And I want to show this to explain that this was not a management issue. The management that Earl and Carol did for their colonies was better than the state average. And yet there was room to improve the survival of the colonies over winter by using the probiotic Super DFM. So that was really encouraging to us, and we continued to collect more information like this and try to collect more specific data on health and well-being and diseases of different control group and Super DFM group that were managed with the Super DFM probiotic. We then started our own apiary in Milwaukee, and our apiary is on the roof of our building. It's an urban apiary of about 20 hives. And we were looking at our bees and the bees in Michigan, and looking at the colony size, looking at frames of bees in the spring. One of the encouraging results that we had was a much better spring buildup and a 30% better brood pattern in the colonies that received Super DFM honeybee compared to the control colonies. We did find that using Super DFM first thing in spring and using it monthly in spring was really helpful for those colonies. We also were looking at Nosema in not the bees infected with Nosema, but just naturally occurring Nosema. We had a study that was done also by Saskatchewan Beekeeper Development Commission, which compared Super DFM honeybee to fumagillin. And they have found that the suppression of Nosema in bees was equal or as good from Super DFM honeybee as in this group that received fumagillin. Oh, and I will be happy to show more information, but I thought it would be really good to have some time left to hear your questions.
Anybody has any questions? Feel free to unmute yourself and ask Vera. I've got a couple questions already written down. Have you ever fed your Super DFM with like dry pollen in pollen feeders early in the spring?
Yes, sir. Yes, we did. So we did feed Super DFM with pollen, and we also took some microbes by themselves and mixed them with the pollen and pollen substitute and dry fed that in the spring. And you can dry feed it separately or together, but we actually have a pretty windy condition. So for us, it's really hard to study that. But in the apiary in Michigan, working with Earl and Carol Hoffman, they do that extensively every late summer and fall.
Okay. We do a lot early in the spring. You can't really open up your hives because it's pretty cold and windy, but the bees do fly every once in a while. And I do feed the pollen, but if I could feed some microbes with it at the same time, I didn't know if sunlight broke it down or if it was still stable. It is protected.
As long as it doesn't get wet, it should be good. You can open feed it just like pollen substitute, or
anyone else have any questions?
Yeah, I have a question, Vera. John Betts. You can't see my face, and that's a good thing. One of the things that I was curious about, I actually used Super DFM here this past summer for the same reasons that I had read that there was an increase in winter survival of about 10 to 20% and potential faster spring buildup. My question is there any longitudinal studies indicating that Super DFM decreases the likelihood of specific brood diseases besides the one you just noticed there at the Nosema? Are there longitudinal studies that show, hey, it also decreases this brood disease or that brood disease? And then the second part is, are there other probiotics besides Super DFM? Thank you.
Questions. So yes. Yes. So I understand your questions, and they're really good questions. So first of all, we're very interested in looking at different brood diseases. The one that we had the most grasp of, just because of the beekeepers that we were able to work with, was chalk brood. Chalk brood was something that we were working with an operation that did about 4,000 nukes every spring, and they had issues with chalk brood. They were happy to test out Super DFM for a couple of months and said that the chalk brood incidence was 70% less in the hive. They split their 4,000 nukes in half or so. And they said that the group of nukes that received Super DFM had 70% less chalk brood. We also have with chalk, with any brood diseases, they don't happen every day in every hive. So we had occasional chalk brood outbreak that we treated and we reported, before and after frame pictures. That's one of the brood diseases. I don't feel confident trying to work with American fall brood outbreaks personally. And I think that the European fall brood, or EFB, is probably the greatest problem in North America right now. So both Canada and United States have seen many years of EFB outbreaks. We are working on finding and testing EFB quickly enough. We find that by the time we collect the samples and get our hands onto an EFB outbreak, at least in our apiary, we don't really see EFB anymore. We did work with a beekeeper in New York who wrote us a letter about using DFM on an EFB outbreak that was happening late in fall, and he had a really good result. and shared all of the information about his hives with us. All of this incremental progress, it does not amount yet to a published paper or a published study that we can do statistical analysis on. So I think that to really kind of get to where we can report a large longitudinal or a larger study that includes many apiaries and many hives, many data points, probably need another 1 or 2 seasons of collecting samples and finding the European fall brood outbreaks near us or somewhere where we can work with. This was, I think that's all that I had to say about the brood diseases. Nosema was actually, it's much more prevalent. So studying Nosema for us was much easier because we could get the samples of the bees, do the Nosema analysis, and then go back and get the samples of the bees from the same hive. And, it was easier to work with than any other disease. So we had pretty good studies with Nosema. Other probiotics, you asked about other probiotics that are available. So we had started, Super DFM in 2014, and we started working with Man Lake. So there is another probiotic that Man Lake makes called Pro DFM. We're not really sure about the quality or any studies that were done with Pro DFM. All of the work that we've done was with Super DFM, and we don't really have anything to do with Pro DFM. So I can't speak to that particular probiotic. We also have noticed that there was a study in 2019 about biopatty, which is like a protein patty or a pollen patty that has microbes or probiotics in it. That's a probiotic that was developed by a group in California that works on human probiotics, and they did a study in Canada. It was a short study. I think it was just 2 hives, but they published it and it looked very interesting. I think they've been talking about it for 2 years, but we don't really have, it's not really available to beekeepers yet. So I'm really curious to hear more about that. Did that answer your question, John?
Yes, it did. Thank you very much.
If there's something that I don't know about, let me know. I'm always on the lookout for new information about probiotics and microbes in general.
I read the other day, and I can't remember where I read it. I thought it was in Bee Culture magazine. There was a study that looks like gut bacteria are a way that bees identify their sisters from the same hive. Were you involved in that study, or is that something you're familiar with? No. Are you aware of that?
I am familiar with that. Yeah, no, I didn't do that anything to do with this study, but that was just fascinating, and I think about it every day. Yeah, I think that was very interesting. So the idea was that just like, say we're using different deodorants, right? As if bees were little humans, we would all smell different, and we would like some pheromones and not other pheromones. And the idea was that bees in different hives have slightly different microbes, and they smell different because of that. And that was so cool.
I agree. And I'm curious how, okay, if you're feeding probiotics, how that might influence that experience for bees.
Yeah, I wonder. So you would need to probably look at splitting and combining the hives or something like that. So if you wanted to answer that question scientifically, you would probably take all sister queens or just, pretty uniform equalized hives and see what happens, if you're combining the hives or if you're splitting them and feeding one probiotic and the other one you're not feeding probiotic. And then they did this very intricate thing, which is a lot of work, and I don't think it's very practical. Like, for example, I'm not going to do that. They introduced bees to each other, or they looked at how aggressive they were to each other. So that would be something that you would do if you wanted to answer this question. On a practical level, we do not see hostility or unfriendly behavior for bees. They still, I mean, even the hives that have all been an apiary that has received the probiotics, they still rob each other if there is an opportunity. So it's not like they, we don't see them, the hives that receive the probiotics be more friendly or less friendly to each other.
Okay.
We occasionally combine the hives and split the hives, but we don't really have any data or observation to show that the probiotics are affecting their behavior. We also work with pretty calm bees, and we try to go for those bees that have this very friendly behavior, so they don't get more aggressive or less aggressive. We don't observe that.
Thank you.
I'll add a little bit here to put full disclosure on this presentation. I went all in on using probiotics, the Super DFM this year. I've been battling the sporadic EFB for 5-6 years with antibiotics. Treat it with antibiotics, it clears up, goes away. Next spring, it comes back. Year after year, I was getting very tired of this. I would talk to Carol Huffman at the bee meetings. That's who really got me interested in this. 2 years ago, I think I did it a little bit, didn't see much reaction. So this year I went all in. My goal was to have 3 doses 1 week apart before May 1st, because around May 1st is when I would see my EFB bloom and explode. I got it to just barely, but a couple hives started with EFB before that. So then I had to make a decision. Do I stop the Super DFM, or do I try to use some terramycin? I rolled the dice. I stayed with the Super DFM. I just kept feeding it on a weekly basis. I had 2 hives that really struggled most of the year. The other hives, after a couple more treatments, they turned around. I never had a hive that would not, with the I'm going to call it a variant. Maybe we'll call it the Northwest Ohio variant of EFB. I was in a talk the other day, and they said, it's just like a cold. Well, I saw this cold kill hives before, not mine, but some new beekeepers that I'd sold nukes to. It wiped them out. I've saw the hives get very sick. So I've always treated, and with the Super DFM, it turned a lot of the hives around, and several of them made as much honey as everybody else. So I think it did help. I will probably know more by spring. Come about August, I'm going to say August, September, October, they probably got 3, most of my hives got 3 more doses going into winter. So I'm treating, I've had good luck with it. So that's one of the reasons I wanted to try to feed it a little earlier with my dry pollen. The EFB I've had has just been a nightmare.
That's fascinating. That's very similar to what some other beekeepers and like Tom in New York has shared with us. And that's why I think that early in the spring, especially when there are recurring problems, is very important to kind of like throw a lot of the beneficial of the probiotic there. I think that it's as the bees go around in the hive, it's not just what happens inside of the gut of the honeybee, but also what happens with the comb and with the cells that the bees are working on. The bees are always doing oral and fecal prophylaxis and eat and chew on hive materials. And that really helps to kind of make sure that there isn't a piece of comb which is contaminated with some of the pathogens. Just by treating maybe like every week, I think that would just cover every corner and every cell in the hive.
Yeah, Carol Huffman told me that's how they did it, and she told me about some EFB things that they were working through, and that's how they did it. So I went in. I was impressed, to be totally honest, because I worked in the green industry for quite a while, and I know there's, I don't want to call this snake oil, but there's always people coming along with things that claim that you spray this on the grass will never die, So I'm always a little skeptic, and I was, would really have loved to have some good studies out there. So I just decided I'm going to do my own study. So that's what I did, was do my own study.
And I think that, thank you. And I think that different variants, like you mentioned, and also, you have to collect the samples of the larva or test the larva. And right now we have variants of EFB that are negative in the EFB test. And that makes it very difficult to kind of collect the data and do the study, especially looking at many different apiaries, because it's a problem of testing and collecting the samples and then making sure that the beekeeper follows the instructions too, right? So doing your own study like that is a great thing. It really helps us to understand what's the best way to use it in different situations.
Larry, I see you posted that you used it this fall for nosema, basically.
Well, there was a study about a year ago. It was in, I believe, in one of the magazines, and they talked about the rotation of the bees during the winter time and whether they're on the outside of that ball or they're more to the inside. Certain positions, which I don't remember, will allow the bees to build up a lot more material in their system before they go out in the spring and do their cleansing flight. In some cases, what will happen is you'll get a lot more splatter on your hive, or you'll get it on your car or your neighbor's stuff, and they'll all complain. And they're saying that it may not be as much as nosema as it is the fact that the gut bacteria tends to be the problem, is they can't really process the way they should. So I basically, for the last 4 years, I treat in the springtime and the fall. In the fall is to prevent, the springtime, and unless you do the study and you take apart the bee and look under the microscope and you know what you're looking for, it may not be nosema. It's just that where their position was in the hive, where they're able to expel, their system at a certain point, that all tends to lead into it. And that's why I just treat both times. I've never had a problem in the springtime with a lot of liquid on the side of the hive or on the cars or something like that. It's been pretty solid, That's what I say, I'll stick to it.
Yeah, that's great. And I think that when to feed in the spring will change a lot with the weather, and same for the fall. But we also think, just looking back to what happens in the hive, those winter bees are made in late summer. So late summer, early fall, I think, is a really good time to make sure that there is enough vitellogenin or fat bodies in the bees. And I think that good nutrition will help there. And of course, then those bees will survive until spring, just like you're saying. And springtime is kind of hard. Maybe I really like the suggestion or the using the pollen feeder and pollen tubes to maybe feed it before it really gets, before you can even open the hives.
Yeah, I agree with Roger on that one too. Yes. Thank you.
We got a question. Do healthy bees have some Nosema spores in them? Is it just not an issue because the bees are healthy?
Yeah, that's a great question. So I looked at hundreds and hundreds of bees and their Nosema spores. And there is a reason why there is a threshold, kind of a treatment threshold, a concern threshold. So when Nosema spores are 1 million spores per bee or more, it really is not healthy, right? So that becomes a problem. You would find, and there are different ways to sample for Nosema. So you can sample one bee at a time and look at each individual bee to see whether or not they have Nosema spores in them. Or you can take hundreds of bees and then look to see if there is like one spore in them. So many times the level of Nosema, the low level of Nosema could still not be a problem, not cause a problem in the hive. So there could be some time, actually there was I think a BA4 partnership. So sometimes you see levels of Nosema that are 10 or 20 times below threshold. And that's really pretty healthy if you look at it generally, especially during the winter. So you look at, late fall, early winter bees in California, they might have, they were able to sample them and they might have like 10 times or 20 times below the threshold Nosema levels. So I would say that's pretty healthy. I also have to say that there are many times where we see zero Nosema spores, like none at all. And that could be in the same hive that had spores in early spring. Usually with every generation and with really good foraging, the bees can have zero Nosema spores, or you would have to search very hard to find the very low level of spores. But many a time that I would look to find any Nosema spores and see like none at all or one.
Do you have any recommendation on how much super DFM to put in like a pound of pollen feeder?
I would know how much to give to one hive, but I don't know how many hives each pollen feeder would. So I guess you can give it like 1 or 2 teaspoons.
Can you overdose them?
No, you cannot overdose on DFM. The safety and not having any side effects was very important to us, and I mean that's part of why we use microbes as opposed to chemicals. But I think that you can use more in the pollen tube. I just don't know exactly how much would be a good number to give you, depending on how much pollen substitute you're using. Another suggestion that I have is maybe using the hive entrance to put the super DFM there when it's too cold to open the hives. As long as it doesn't get wet, like it's not raining, I think that might work too. That's one of the ways that we have been putting quickly the probiotic in the hive. And this way you can do like 1 scoop or 1 teaspoon to every hive.
And a question, when do you put it out in the spring? When I put out pollen, I'm going to answer that question as yes. I try to get it out as early as possible. The first time I inspected the hives in the spring, I had the bag with me and start applying it. We have another question of how do you normally apply? Sprinkle on the frames. Yeah. And that's, I see Vera's nodding her head. Yeah, I have a tablespoon I scoop it out of the bag, level it off, and I sprinkle it along on top of the frames. And the only reason I know what it looks like is because I used to watch Miami Vice when I was growing up. And it looks like lines of cocaine because it's white. And you sprinkle a little, I tried to sprinkle it, lay it on the top bar so they can come up and eat it. And yes, they do eat it just like Vera's picture. They start gobbling it up. You'd swear it was powdered sugar the way they go after it. But I sprinkle it along there, and I try to do it on a non-windy day. Sometimes the wind blows it in and blows it on the bees and in the comb, and I figure the bees eat it out then. But it, honest to God, it looks like you're putting lines of cocaine on it.
Yeah, it's so we use sucrose as a carrier for our microbes, and that's because the bees like the taste of sugar, of course. But it's very important to us that it doesn't, doesn't smell bad and doesn't make something that is unappealing to the bees. Yeah. Yeah, we usually put it on the top bars too, but sometimes we put it through the entrance.
Well, I see Joe Kovaleski's here, Peggy Garnes is here. We've beekeep long enough that my bee inspector, when I first started beekeeping, told me that's how you applied the terramycin to prevent EFB and AFB, is you would just scoop it up and throw it in the front entrance of the hive. That way, especially in the fall, if it was getting cold, you didn't have to open the hive, you just pitch it in. And we were told to treat twice a year, or 2 treatments, one in the spring, one in the fall, to prevent these brood diseases. And it was very much common knowledge just to treat prophylactically. I had when I started treating for nosema with fumagillin and learned that fumagillin killed all the good and bad bacteria in the bee's gut. And I'm going, well, how in the hell do my bees survive winter when they're going into winter with no gut bacteria in them? And that's kind of where my light went on years ago. I quit feeding fumagillin. I figured I'll take my chances with the nosema because I never really had nosema problems. I'll take my chances with that. And because I knew I was screwing up the bees' guts with the fumagillin, especially real late in the fall where they didn't have time to readjust going into winter. So that was the first one. And the other one, I got away from the antibiotics prophylactically a long time ago too.
And I thank you so much for understanding that, because you're right, like many years beekeepers were taught to use antibiotics prophylactically, right? And I'm really glad to see that we're moving away from that. It's just not good for the bees, and it's also not good for us to eat the honey or use anything from the hive that has that could have a trace of antibiotics too. And how do bees survive without bacteria? So I mean, just like I said, there's just a ton of, nothing is sterile in the hive. Everything in the hive and out in the field is covered with bacteria. So whether you're talking about bee bread, comb, pollen, anything on the flower, nectar, it all has plenty of good bacteria in it. If you keep erasing and wiping out bacteria with an antibiotic, new ones will come in, right? New ones will always come in. But part of the problem is that when the new ones come in, let's say if I delete all my Facebook friends and I make new Facebook friends, I could end up with too many bad apples. And the same thing happens in the bee gut. If you keep cleaning it out, every time you clean it out, there's a chance for the bad guys, for the pathogenic bacteria to get out of balance, to have a little bit more sway and to cause the diseases.
This is Joe.
Sure.
I got
I'm sorry.
a cute little way that you can take this out instead of taking your bag and a scoop. You got an idea of how much you're going to put on the top bars. You take one of these little 1 pound squeeze jars and this little pointy cap, and you fill that up with your DFM, and you just go to your hive and you go, psh, 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 and it works just slick. You don't have to worry about dipping a scoop in a bag and carrying the bag around. Just fill your little bottle up and it works just slicker than Dickens.
Oh, I love it. That's a great way.
Thank you, Joe, for the recommendation. I think that will make my life easier.
Yeah, you know, and again, like Vera said, you know, you don't have to be precise down to the microgram. You know, if you get a little bit more, it's not going to hurt them. But you know, when you know about how much you're going to put on there, and it works just slick.
I kind of, half a teaspoon for a little nuke. If it was a big super hive, maybe it got a teaspoon in or a tablespoon and a half. I just kind of judged it on the size of colony too.
I have a question for those, because I've only used it once, like I said, and it was late summer, early fall, and I cannot remember the directions, but I remember when I read the directions at the time, I thought it said something to the effect that don't feed this with like pollen patty or pollen or whatever. Does anybody remember that? Am I misunderstanding or it? Because I remember when I put it on, I thought, oh crap, I already put the stuff on 2 or 3 days before that. And I thought it was a pollen patty. And I thought, did it say, are the directions that you're not supposed to like put 2 different things on? Just help me protocol wise, because I can't remember what I read 6 months ago.
Yeah, so you're pointing out that we need to be more clear, because using it at the same time as a pollen patty or any feeding is not a problem at all. We just don't want it to be mixed into the pollen patty, into the wet pollen patty that you make, 2 or 3 days earlier, and it sits, and then you bring it to the hive and the bees consume it over a week. Keeping the microbes wet like that could be a problem, or putting it into sugar syrup that you're boiling and then it sits out in the sun. We just don't want it to be mixed, like touching each other. But as long as it's not touching each other, say I have my pollen patty over here and I have my line. Sorry, it's hard to snap out of it. And I have another bar that I put the spoon of DFM on. As long as it doesn't get really wet. And the reason why we also don't want to mix it is because when you just sprinkle that spoonful of DFM, they consume it pretty quickly. Whereas for a big pollen patty, they might be working on it for days. If it's a really booming hive, they might eat up that pollen patty and the paper with it. But sometimes it takes days to eat it, and that's why we just don't want them touching each other.
Thank you.
We also know that some beekeeper there's going to be a situation where you have to use the antibiotic. And if somebody does have to use the antibiotic, we don't want that to be done at the same time as you do the probiotic, because that's probably not going to help. So maybe if there is an antibiotic treatment, finish that first and then start the probiotic. Mm-hmm.
We had another question is, does it have a shelf life or an expiration date?
Yes, it does. Yes. So we have been doing it for 5 years when we, so our shelf life is 5 years, and you do need to keep it in a cool and dry place. 5 years is the shelf life on the product.
I was told to keep it in the freezer. Is that true?
On the worst, it's very good. Sometimes you don't have a freezer space to put it. So if you just keep it in a cupboard, that's fine too. An important thing is if it's an open bag, we have a zip seal so that it stays dry and nothing can get in there. So zip it up, and you can put it in the freezer, or you can keep it in the freezer altogether, or you can just keep it in the cupboard as long as it's not really hot. Truck bed, for example, could be a problem.
I'm just looking to see if there was any more questions.
I really like the questions, and I think that the question that John asked really helps me to go back and say, we need to make sure that we are clear, because I think that as we were new to beekeeping, our instructions didn't necessarily translate that well into practical beekeeping life.
That's good. Good job, John. Because I know I must have skipped that line in the directions, John. And because I know I had pollen patties on and sprinkled it between the pollen patties and stuff like that, especially going September, October. So does anybody else have any questions they want to type in, ask Vera?
I'd be happy to share my contact information too. So if you guys have any questions or anything like that, or if you want to do a really cool study and collect some data with Nosema or EFB, we can talk about it. I'm always happy to work with beekeepers.
Okay. Joe, I see you unmuted yourself. Do you have anything you want to add to? You must have
No.
used it for a while.
Yeah, I've been using it for pretty much since it first came out. You know, I know Earl and Carol through being master beekeepers. And when it first came out, I used it on a hive that I had a very bad case of chalk brood, probably the worst case I've ever seen since I was keeping bees. I had an entire side of a frame that was completely full of chalk brood mummies. And I put the DFM on there, and within 2 weeks, I did not see any chalk brood in that hive after that the rest of the summer. And it's like, so I can attest that, yes, it does have some effect on chalk brood in hive. And like I said, it cleared up and never came back. And we all know that a lot of times when you have a bad case of chalk brood, it's going to keep resurrecting itself throughout the rest of the summer. Periodically, you're still going to see it. And that cleared it up. And did not see any signs of chalk brood the rest of the year in that box. You know, so I can attest to, yes, it does help out with the chalk brood. So, and other than that, I'd just like to say thank you for letting us in, and thank you, Vera, for your presentation.
Oh, no problem. I appreciate all of, like, I've been to many of the Medina County ones, and I knew we were going to probably have extra space here, so might as well invite as many people as possible. So you're welcome. We got several thank yous. Vera, people are thanking us for this.
I'm really glad to talk to you. I think every time we talk and I talk with any beekeeper, there's 10 new ideas. And one thing that I wanted to say is that, I mean, obviously there are serious problems, and sometimes, you know, the more serious the case of chalk brood or fall brood, the more obvious or the more you need to use, for example, super DFM. But there are also healthy bees. There are bees that are healthy, that are booming, that are thriving. And that's another thing that I'm really interested in looking at, because as we learned beekeeping, we learned how to do queen rearing, and looking at, you know, queen acceptance rate and queen health is also such a fascinating, interesting topic. And just listening to Joe talk, I thought about that too, that's a whole, you know, that's a whole chapter of beekeeping, is how do nutrition and microbes in the hive, how do they affect how long the queens live, and how good they are in the end. So that's just another thing. But obviously it's if your bees are super healthy, I hope that they are always, you might you might not notice chalk brood or fall brood, and that's great. And I hope that you don't.
I guess, you know, what really, you know, I'm always thinking of how to make the bees healthier. And we all know that nutrition's lacking versus, you know, we don't have enough diversity in our flowers. There's not enough of them. So anything to make them healthy. I mean, if the way they push probiotics on us, on the human diet, that's not being eaten properly, I'm sure the bees are even further behind. And that was part of what I was thinking about with with my EFB is if it's being a gut issue and if I can make the guts healthier somehow, it's got to help me. So.
You're absolutely right. You're absolutely right. And it's true that nutrition, in terms of the forage and the flowers that are available, continues to be a very important thing for beekeepers. And I think that during the times of dearth, during the times when there supplemental feeding is needed and you need to feed pollen and you need to do pollen petties or other feeding, only doing the probiotic is not going to feed the starving bees, right? It's not a food. It's just something that helps nutrition and digestion. So that's an important reminder because, like you said, sometimes we might not be, I want to be very transparent, it's not food by itself. It's just the microbes that you still need to feed the protein supplement to your bees. Just feeding the probiotic supplement is not going to feed the starving bees.
Right.
So if your bees are in a pollen dearth, you need to feed pollen or protein substitute. Just putting the probiotic on the hive that needs pollen is not going to help very much. So just want to be clear because I don't want to make any promises that are unrealistic because I am very much aware of claims and promises that have nothing to do with reality. And I want to make sure that we only talk about things that are understandable and have evidence to support them.
Maybe that's what I should add. It worked for me. I don't know if it's going to work for everybody. That's my second disclaimer is I've tried it. I like it, but I won't guarantee it works for everybody. So Mary Nelson, you're a salesperson of this. Do you wanna speak?
Roger,
Yes, Emma.
do you know any local vets that do bees in just in case I ever need one?
That is a very good question. And to be honest, as far as Fulton County, no, I'll have to look in my notes. There is a vet in Sylvania that trusts our bee inspector. So when the bee inspector calls, fills out the paper, comes to your house, finds you have, let's say, EFB, you need some terramycin prescription, he'll call the vet will write you a prescription for it without looking because he trusts, but that's been a very big issue. I know you're new, but when they made vets, no vets had any experience with bees. They weren't taught insects in veterinary school. So they are getting some. I sat in on a bee vet conference out of Canada, and I even got a certificate that says I attended. So don't know what it's worth, but it is very difficult, Emma, to find a vet that will do a prescription for bees. So,
Okay, thank you.
You're welcome.
Mhm.
And that goes for everybody. It is tough.
So
Mary?
Roger, yeah, hi.
Go ahead.
So I've been selling Super DFM for over a year now and sell to sideliners, hobbyists, commercial guys that will buy big drums of the product. So anyone interested in buying Super DFM, you can contact me directly and I will give you a 25% discount on your first order. And I also will give you a thank you gift. This is just because it is a consumable product and you'll probably be needing more. So introducing the product to you should be a good experience. I'm sure that just from the testimony you've heard today, that it's very likely your bees will benefit from the Super DFM. So I'm on Facebook. I can also put my phone number out to everyone. Contact me by text or call. And if you have any questions about, anything, I can help you. I'm not a beekeeper, but I work with beekeepers and I love bees and I love beekeepers.
Very good. Thank you, Mary.
You're welcome.
Emma, the vet in Sylvania is Countryside Animal Hospital, and it's Dr. Pfeiffer. I think it's Paul Pfeiffer. So if you want to write that down, hopefully you never ever have to call a vet for your bees.
Right.
Can you repeat that?
It's Countryside Animal Hospital, and it's Dr. Pfeiffer, Piffer on Whiteford Road, Whiteford and Monroe Street. Thank you, Tony, for pulling that up quick for me. Any other questions for Vera? Well, Dr. Vera, I appreciate your time. Thank you very much. It's glad to have you.
Thank you. Thank you so much, Roger, for this conversation. This was a great discussion. Thank you.
You're welcome. Thank you.